Hello everyone, and a very warm welcome to our webinar on analysis of Lexogen's QuantSeq data using Partek Flow. We will start the webinar with an overview of QuantSeq technology and data analysis. Then we will briefly introduce Partek Flow, while the main part of the webinar will be dedicated to a live demo of analysis of QuantSeq data in Partek Flow. At the end, it will be your turn to ask anything on the webinar content or our analysis solution in general. Without any further ado, it is my pleasure to introduce Dr. Stephanie Bannister from Lexogen. Stephanie is an application scientist at Lexogen. She provides technical and application support to Lexogen customers and develops technical resources for Lexogen RNA sequencing products. Stephanie received her undergraduate degree in biotechnology from Deakin University, Australia, and her PhD at the University of Melbourne, Australia. Her core scientific experience encompasses molecular biology with a focus on microRNA regulation, RNA interference, and genome editing, and reproductive biology in chicken embryos and marine polychaete worms. Our second speaker today will be Dr. Ivan Lukic, a senior field application scientist from Partek. Ivan obtained his MD and PhD in immunology from Zagreb University School of Medicine in Croatia and moved on to postdoctoral training at the University of Heidelberg in Germany. As a research scientist, he authored or co-authored several dozen manuscripts on multiple roles that the immune system plays in health and disease. Ivan joined Partek in 2011 and has since advised hundreds of Partek users across the globe on various aspects of experimental design and data analysis, helping them to present their stories to the world. Stephanie, off to you.
Thank you, Paul, and thank you to the organisers for organising this webinar. Hello to everyone who is tuned in today. As Paul mentioned, my name is Steph and I'm an application scientist at Lexogen. Tonight I'll be talking about the QuantSeq 3' mRNA-Seq library prep kit technology and data analysis. Some of you may be familiar with QuantSeq, but I'd like to start with an overview of the QuantSeq technology and protocol. So the QuantSeq 3' mRNA-Seq library prep kit is specifically designed for expression profiling RNA-Seq projects. Let me first start by briefly describing how expression profiling sequencing differs from whole transcriptome sequencing. Whole transcriptome sequencing, as shown on the left, is designed to generate reads that map across the length of the transcript. It is widely used for mRNA-Seq and total RNA-Seq sequencing projects and is ideal for performing de novo transcriptome assembly or isoform detection and quantification. With whole transcriptome sequencing, the read data must first be normalized by transcript length to give FPKM values for quantification. On the other hand, expression profiling sequencing, shown on the right, is a simplified approach, ideally suited to studies that aim to profile gene expression levels. For expression profiling, only one fragment or tag is generated per transcript, and this can be used for simple counting approaches to identify transcript abundance. The QuantSeq 3' mRNA-Seq library prep kits provide a method for preparing libraries for this kind of expression profiling sequencing. As the name suggests, this is a 3' mRNA-Seq method, meaning that one tag is generated at the 3' end of each transcript. To achieve this, an oligo-DT primer is first used to prime reverse transcription. This binds at the start of the poly A tail of the mRNA and contains a partial P7 adapter sequence. After reverse transcription, the RNA template is removed and random primers are annealed to the first strand cDNA, which contain a partial P5 adapter sequence at the 5' end. Once these primers anneal, they are extended to generate second strand cDNA. The double-stranded cDNA library is then taken onto library amplification, where using PCR, the complete sequencing adapters are added and unique I7 and/or I5 indices can be added for multiplexing. The whole protocol is very streamlined and takes only around 5 hours to prepare ready-to-sequence libraries, with around 2 hours of hands-on time. So now I will give you a quick overview of the data that you can get from QuantSeq libraries and how this can be used for expression profiling. Here you can see two graphs. The graph on the left is showing a plot which compares the read coverage for QuantSeq libraries, shown in green, to whole transcriptome mRNA-Seq, shown in grey. As you can see, for QuantSeq in green, the majority of the reads map to the 3' end of the transcript, while for the mRNA-Seq, The reads are distributed across the length of the transcript. For this data, the ERCC spiking controls were also included in the library preps. The read coverage for the ERCCs is shown by the grey and blue lines, respectively. The graph on the right-hand side is showing the results of differential expression performance, comparing QuantSeq with the filled circles and to whole transcriptome mRNA-Seq for the open circles. For this experiment, the ERCC X-fold spike-in mixes 1 and 2 were added to 2 different sets of libraries for each library prep method. This was designed to achieve 3 expected fold change ratios. While this plot is a little bit complicated at first, it shows that the QuantSeq libraries with the filled circles are having a higher true positive detection and lower false positive detection rate than are shown for the mRNA-Seq libraries. This indicates that the QuantSeq library prep method provides stable and robust gene expression quantification, also for differential expression analyses. So QuantSeq libraries are also well able to detect and quantify low abundance transcripts. Here in the left-hand plot, you can see an input-output correlation for the 92 ERCC spike-in controls that were included in a QuantSeq experiment. The correlation is very high at around 0.972, which indicates that the expected relative amounts of each ERCC added to the libraries were correctly detected in the final sequencing reads. The graph on the right-hand side is showing the performance of QuantSeq compared to microarrays. This plot shows the number of detected genes on the y-axis for different levels of expression values shown here as minimum detection value. Here you can appreciate that the QuantSeq libraries shown in green are giving a higher gene detection level at lower gene expression or detection values than you can see for the Illumina arrays, which are shown here in blue. This also demonstrates that QuantSeq is a highly ideal alternative for gene expression profiling compared to microarrays. So let me now summarize the key features of QuantSeq. As you can see, it's a highly cost-effective method, costing only $19.80 per prep. It provides transcriptome-wide analysis of gene expression with exceptional reproducibility and sensitivity for low transcript detection. The protocol takes only 5 hours with only 2 hours of hands-on time, and we provide 96 I7 and 4 I5 barcodes that allow for unique multiplexing of up to 384 samples. What I didn't yet mention is that this protocol is able to incorporate low amounts of total RNA, as low as 100 picograms, and the protocol is also highly suitable for low quality and FFPE RNA samples. As a final point here, and because today we're talking about data analysis, because QuantSeq generates just 1 3' tag per transcript, this greatly simplifies the data analysis process, which allows researchers to analyze their own RNA-Seq data, even if they haven't got any significant bioinformatics experience. So now let's move on to the QuantSeq data analysis pipeline. For any RNA-Seq data analysis workflow, there are essentially 3 main steps. The first step after receiving the raw reads in the form of a FASTQ file is to perform trimming and quality control. The second step is aligning those trimmed reads to a reference genome, and the third step takes the aligned reads and processes them for gene counting to extract gene expression levels. For the QuantSeq data analysis pipeline, our Lexogen bioinformaticians have put together a simple analysis pipeline that optimizes the data analysis of QuantSeq read data encompassing these 3 core steps. As the transcript fragments originate at the 3' end of mRNA in QuantSeq, we include trimming of poly-A tails and low-quality sequence as well as adapter sequences. This helps to improve the downstream mapping rates. Before and after trimming, the pipeline also generates quality control reports to allow for analysis of read quality. Once the reads have been trimmed, they then get taken to alignment. We align the reads to a reference genome, and as it can make a difference to the mapping results, it is best to always use the most up-to-date reference genome annotation available for your species. For our ideal analysis pipeline, we use the star aligner, and we have included additional parameters that maximize the unique mapping rates for QuantSeq data. Once the reads are aligned to the genome, the pipeline also generates alignment quality control statistics, and then the aligned reads are taken and processed for gene counting. For QuantSeq data, we define mapped reads for gene counting as those that are uniquely mapping to the exons of the gene body, and we use HT-Seq count to extract the read counts per gene. Those read counts can then be used for downstream differential expression analysis using various tools. So actually, this whole data analysis pipeline for QuantSeq can be performed using freely available analysis tools. However, accessing and running those different tools individually can still present a challenge for researchers who haven't got much knowledge of bioinformatics. This is where Partek Flow comes in. By integrating this pipeline into the user-friendly data analysis interface on Partek Flow, QuantSeq customers can easily access and run this pipeline to analyze their QuantSeq data, even if they have no previous experience with bioinformatics. All of the required steps of data analysis are performed with an easy-to-navigate interface, and Partek Flow also provides access to a range of options for visualizing the results of their experiments. I will now hand over to Ivan from Partek, who will take us through the use of Partek Flow for QuantSeq data analysis.
Thank you, Stephanie, for this great presentation. Good afternoon or good morning, everyone, depending on your time zone. This is now Ivan from Partek speaking, and please permit me to welcome you to the webinar. Let's start by an introduction to Partek Flow. So, what is Partek Flow software? Partek Flow is our package for comprehensive and powerful genomics analysis. Once installed on a cloud, compute cluster, or a server, Partek Flow can be accessed from anywhere in the world using a device with the internet browser. All the analyses are performed via an intuitive graphical user interface. Partek Flow supports numerous genomics workflows and, among other features, facilitates setting up collaborations across multiple centers. One of the supported applications is RNA-Seq analysis of Lexogen's QuantSeq data using a streamlined pipeline that was developed through collaboration of Lexogen and Partek teams. The pipeline takes FASTQ files, removes poly-A sequences and sequencing adapters, performs quality-based trimming, aligns reads to any genome, and then quantifies the reads to any transcriptome database. Furthermore, quantified reads can be pushed to differential expression analysis, followed by biological interpretation. And now let me switch to Partek Flow and show you all those steps live. This is Partek Flow, and let's start this walkthrough by a visualization of QuantSeq sequencing data. In this example, I am plotting sequencing reads of 3 experimental groups as histograms. There is the control group at the top in blue, and 2 other conditions in this experiment, drug A in the middle in red, and drug B, lowest track, in orange. As we have just heard from Stephanie, the QuantSeq kit interrogates 3' ends of genes, and this is exactly what we see here. Sequencing peaks aligning nicely to 3' end of the gene shown here at the bottom. And now let me show you how easy it is not only to get browser visualization like this one, but to perform start-to-finish analysis of QuantSeq data in Partek Flow without advanced bioinformatics skills. To start with, I will show you how to import your sequencing data to Partek Flow. For this, I will also need to create a new project. So projects, create new project, and give my project a name, for example, QuantSeq Analysis. Created. I am now in my brand new project, and you see a message that I need some files before I can run an analysis. Therefore, import data. As you see, we have different wizards to help you handle various multi-omics assays and scenarios. For QuantSeq data, I will go for the first option, automatically create samples from files. The next page is the file browser. Depending on the location of the data, I will use the respective option here under Select Files From. In this case, the data is on the same computer as Partek Flow, and we are exploring the directory structure in the panel on the left. The browser window on the right shows only types of files that are supported, which is a very helpful feature. Okay, I have here 7 files selected, and 6 of those correspond to my 6 experimental samples. I will uncheck the first text file and proceed with FASTQ files only. Note that I do not have to uncompress the files first, because they are currently in a compressed format, but Partek Flow will do it for me on the fly. We'll come back to the first file, the txt file, in just a few moments. Right now, let's create samples. There is some processing going on in the background, so I will use this time to add attributes to my samples. My sample attributes are organized in a file, which is particularly useful for clinical research, where metadata can be quite abundant. Therefore, assign sample attributes from a file. Now we went back to the same directory and the file browser, and it is time to use the txt file called attributes.txt. Select, and next. This is the preview of my attributes file. As you recall from the browser view, there are 3 groups in this experiment: control condition and then 2 drug treatments. Partek Flow automatically recognizes sample IDs on the first row and sample attributes on the second row, so there is nothing for me to do here but push import button. Back to the data tab, my samples are now annotated. The first 2 samples belong to the control group, while the other samples were treated with either drug A, rows 3 and 4, or drug B, the last 2 rows. I'm now ready to start the analysis, and for that I will switch to the Analysis tab, where action actually takes place. Here it is, Analysis tab. This is what Analysis tab looks like at the beginning of a new project right after data import. All the steps of the analysis are represented graphically by symbols on the canvas. We call them nodes. To work with my unaligned reads, there is no need for command line. I simply select the node, and the toolbox appears on the right. The toolbox is context sensitive and only shows me those tools that can be invoked at a certain stage. In this way, software helps me to pick up an appropriate tool at each step. Typically, what I would do here is first launch the pre-alignment QA/QC tools to assess the quality of raw reads, then evaluate the reads, and decide on any processing of the data prior to alignment. The respective tools are here in the pre-alignment tools section. Luckily, I do not really have to do that right now. We work together with Lexogen team to add a pipeline for streamlined analysis of QuantSeq data in Partek Flow, which will guide me through the initial stages of RNA-Seq analysis and lead me to a clean data set which is ready for biological interpretation. All that with just a few mouse clicks and a few decisions relating to pure biology and not bioinformatics. To add a QuantSeq pipeline to the toolbox, I will use this option here in the lower left corner, Import a pipeline. The pipeline itself is hosted on Partek website, and there it is right at the top. Here's a quick preview. The pipeline includes the steps that were discussed earlier in the webinar, so I can close it, close the preview, and go import the pipeline. Okay, I came back to my project, and now when I select Unaligned Reads Data node, there is the pipeline section in the toolbox. Let me open it. Yes, the Lexogen pipeline is inside, and I just click on it to launch it. First, I need to select the file with sequencing adapters. As you see, the file itself is not hardwired into software, and if needed, an alternative file can always be specified. Let's browse to look it up. Here's the file browser and my adapters file. Continue. Next. Okay, what reference genome do I want to align to? The data I'm working with is human, and I want to use the build AG38. Next. And third, what annotation should Partek Flow use to quantify the aligned reads? We support all the major databases, and you can in addition add a custom annotation. For this presentation, I chose the current RNA-Seq build, you see it on the screen, and click on Finish. Here we go. My analysis is now in progress. I can simply lean back, relax, and wait for the confirmation email from Partek Flow that the pipeline has completed and that my gene counts, here on the right, are waiting for me. As you know, this can take some time, so let me show you the downstream steps in a project that I already analyzed. Okay, projects drop-down list. And this is the project that I want to show you, QuantSeq RNA-Seq. Oops, I was sure I clicked on it. No, I didn't. This is what a mature RNA-Seq analysis looks like, and Lexogen pipeline that was just discussed covers the first 5 steps from unaligned reads on the left to gene counts here in the middle. In addition to the processing tasks, and you see them on screen, trimming adapters, trimming bases, aligning reads, and quantifying to annotation models, we also provide detailed QA/QC reports so that you can monitor what is going on with your data at each stage. For example, I will open the pre-alignment QA/QC task after the last round of trimming, that's this one, so that I can inspect the data that is being pushed to alignment. Open the task. This report starts with an overview table. Each row is a sample, while columns contain typical QC metrics. There are also 2 diagnostic charts showing average base quality score per position on the left and average base quality score per read on the right. Each line on these plots represents a single sample. Alternatively, to focus on a particular sample, click on the name of that sample in the sample table. Again, header contains summary, while these charts below provide detailed diagnostic information. Going back to the project, Partek Flow is not a black box, and we provide all the details on each task. As you know, the Lexogen pipeline delivers gene counts data node, which is right over here. To find out more on the upstream procedures or on the steps leading to the gene counts, I will invoke data summary report on the gene counts node from the toolbox. This report is a great audit feature, and you can use it as an electronic lab book. Everything is neatly documented. For example, at the top, we see input data, who imported the data and when, and what is the total file size. And then we go step by step. The adapters trimmed, poly A tails, then bases were trimmed, reads were aligned, and quantified to an annotation model. Show Hide Details link under each task lists all the options that were used with that task. Let me use Align Reads as an illustration. We see all the aligner options that were applied in this run, and any non-default settings are highlighted in red. These specific settings are used here as recommended by Lexogen scientists, we heard this before, to maximize alignment rates. And what if you want to try out something different, like another aligner? We are not locking you in. You simply start with Trim Reads Data node and pick another aligner from the toolbox. You see the full list here, and in addition to the star aligner, Tophead 2 or GeneSnap can also be considered for RNA-Seq projects. Okay, and how do you proceed with your RNA-Seq once the pipeline produces gene counts? Although quantification results in both gene and transcript level output, QuantSeq data should be analyzed on gene level only. The data in the gene counts node is raw, so I want to normalize them. Toolbox, normalization and scaling, normalize counts. There are many normalization algorithms available in Partek Flow, and you can go for a single one or combine a few. When analyzing this data, I actually use the upper quartile normalization. Okay, the result is in the project. Over here in the normalized counts data node. Let's take a look at the report. The table at the top contains summary statistics of the normalized data, and we also see the transformation that was used. Personally, I like the charts better because I can immediately see the effect of the normalization. The expression levels per sample are summarized using box and whiskers. The plot on the left shows data before, and the plot on the right shows data after normalization. We see the effect here, nice and smooth data distribution. I'm happy with the outcome of normalization, but sometimes you may want to compare different transformations. For that, we have a very cool feature available. What I can do is select the current normalized counts task node, which is this one. Go to the toolbox and open task actions. Here, this option, rerun with downstream tasks. It will allow me to pick another normalization and leave all the downstream procedures as is. In that way, I can easily compare the effect of normalization on my results and decide on the final pipeline for my project. Okay, once the data is normalized, it is time to perform exploratory analysis. For this, I used principal components analysis. This is the result. Each dot on the plot is a single sample, and the experimental groups are annotated by color. The plot is interactive. I can rotate it, zoom in, or zoom back out. The image file can be exported using the save icon and then used for publication or work in progress report. There are many customizations that I can perform. For instance, highlight the groups like this. The gene expression pattern here indicates nice separation between the conditions, meaning that the two drugs lead to distinct changes in transcriptional profile, so my next step is to detect the differentially regulated genes. Now I will start with my normalized counts, go to the toolbox, and statistical tools are here under statistical analysis. I will now invoke GSA, which is our award-winning algorithm. The first dialog allows me to specify sample attributes that should be included in the model. I have one in this experiment, which is condition. Next, I will then use the second dialog to focus on specific contrasts. What I want to compare is drug A versus the control, add comparison, and then drug B versus the control. But I can also combine the two conditions on the fly as if they were one group, and again, contrast versus the control. Finish to start calculation. The result of statistical analysis is presented as a table, as a gene table, which is here under feature list data node. So here's the table, genes are on rows, and statistical output on columns, organized per contrast. So this is the first contrast, then the second one, drug B versus the control, and the third contrast, both treatments versus the control. For each contrast, we see a raw p-value. If you take a look at the columns, false discovery rate corrected p-value, ratio and fold change, as well as group means. Now, going further, visualizing significant genes per contrast is very easy. Just click on this icon here, and it opens the volcano plot. Fold changes on the horizontal axis, p-value on the vertical axis, and each dot on the plot is a gene. Upregulated genes are shown in red and are here in the upper right quadrant, downregulated genes in green in the upper left quadrant. Alternatively, to detect significant genes, I can use the filter panel on the left-hand side of the gene table. The table now contains just over 9,400 genes, and this includes significant and non-significant genes alike. Let me first apply fold change filter with a cutoff -2 to +2. You will notice that the number of results change to reflect the filtering. I I can add additional filter, for instance, p-value, and use the cutoff 0.01, which leads me to the final list of 10 differential irregularity genes. Now, I will push only those genes to the downstream analysis by hitting the Generate Filter Node button. You see it in the lower left corner, which actually extracts my significant genes to another node. This one here, feature list after filtering. This is also a gene table, but with 10 genes only, my significant genes, and the filter criteria are listed in the top left. Now let's take a closer look at one of the genes, for instance, this gene here, almost in the middle on the 6th row, CP83. In spite of this gene's very low level of expression, we see the total number of counts, it was captured by QuantSeq Kit. To find out more, I will click on the dot plot icon here on the left. It opens the visualization window. Each dot is a single sample, and the samples are organized per groups. The chart is also interactive and can be extensively customized. For example, I can reorder the groups, or I can add box and whiskers on top of the chart. This chart also shows us that QuantSeq Kit not only detects low abundant genes, but in addition enables confident statistical analysis of those genes. Even at this level of expression, we can clearly tell the difference between 3 experimental conditions. Let's go back to the project and go to the list of significant genes. Another section of the workflow is biological interpretation. Here's the idea. Looking at my significant genes, I would like to know whether the list of genes is dominated by a particular pathway or gene group. To find it out, I can use KEGG enrichment analysis to look for overrepresented pathways, or enrichment analysis to look for overrepresented gene ontology groups. The results are organized in a similar fashion, so I will open gene ontology enrichment result. It's here. Enriched groups are on rows, ranked by the enrichment score, and basically the higher the better. The top group is hemoglobin complex, and by looking at the gene breakdown table on the right, I can tell that the group consists of 11 genes, 2 of which are present in my list of significant genes, while 9 are absent. So what are those 2 genes? View extra details button, and they're right over here, genes in list. And of course, I can plot the transcriptional profile per group using a heat map. From the list of significant genes, I go to the visualizations, hierarchical clustering. Cluster controls are very flexible. We can cluster samples or genes or both and work with different clustering metrics. I actually use the defaults, and let's take a look at the result. Nice heat map, samples are on rows, genes on columns, and expression levels are shown by the colors: up-regulated genes in red, down-regulated genes in green. You know by now that all our charts can be extensively customized. For example, let me first change the color scheme. Instead of green, I want to depict the down-regulated genes in blue. Like this, controls are on the left, pick blue, and OK. We see the sample IDs on the right, and instead of those, I want to annotate the chart by group labels. I'm actually going to remove sample labels and then overlay attributes on the chart. Finally, the order of the branches in the dendrogram is arbitrary, and I'm allowed to flip around branching points. We can do that easily using the flip mode. And here we go. Once I'm done with the customization, I click on the Save icon and export this image. Going back to the project, we have actually reached the final stage of the analysis, or to rephrase, I have just shown you how to use Partek Flow to perform a start-to-finish analysis of Lexogen's QuantSeq data in an easy and efficient way. To wrap up, Lexogen QuantSeq kit with Partek Flow software deliver fast, easy, and comprehensive gene expression analysis. You can start with raw reads and take your data all the way down to biological interpretation without advanced bioinformatics skills. Thank you for your kind attention.